Hello everyone, welcome to the 9th talk of CAPS Talk series. So this is the last talk from protein structural parts, and today we'll be plunging into protein modeling. Over to you, ma'am.
Yeah, thank you. Thank you, Shaili. I trust you can see the screen. All right. So what we have been doing in the past few weeks is to appreciate domains, domain swapping, and then we also discussed structural analysis that can be pursued with domains such as being able to align distantly related domains together and then looking at the residue pairwise and higher order interactions. Yes, and that has implications and applications in many areas, such as modeling, validation. So we discussed the harmony as well. But what we will be discussing this week is actually going into the tool, which is homology modeling or comparative modeling. These are just used interchangeably. And in our lab, we use this modeling as a tool. We don't research in the area, but we use this as a tool to then ask biologically relevant questions, which means we can go from one biological system to another. So what we will try to do today is to present the applications of homology modeling by taking you through 3 different stories. And the first of them is a toll-like receptor. The toll-like receptor has 3 major components. The ligand binding part, which will be in the extracellular milieu, and there will be a transmembrane helical region. And following that, we have an intracellular domain, which is, of course, involved in the consecutive signal transduction. And what we had noticed is there are many players in the toll-like receptors, and you will hear it from the speakers soon. And one of the aspects is there is an intracellular domain, which is called as a TIR domain, which has to be present as a dimer, because the whole toll-like receptor dimerizes upon ligand binding and so on. So therefore, the TIR intracellular domain has to be a dimer. Also, other thing to appreciate is some of them can directly participate in signal transduction, whereas some others require what are called as adapter domains, which enable them to go into the stream of the signal transduction itself. So one of the adapter domains, which is contained in a gene called TRIF, we got interested, and one of my students, ex-students, Mahita, particularly spent some time modeling the TRIF, and particularly how the N-terminal domain would interact with the TIR domain, and why they would interact, because it forms a level of auto-regulation or auto-inhibition, as it were, where the N-terminal domain, as long as it's stuck with the TIR domain, it will not let the signal transduction machinery to process. A very beautiful set of regulation by nature. But then what Mahita has done is to model the individual domains and also try to model how these 2 domains will be stably talking to each other. Not only there are hydrophobic interactions, there are electrostatic interactions, and they remain as witnessed through molecular dynamics. And then the TIR domains themselves have a very interesting profile of dimerization, because some of them dimerize, let's say, in this way, and some of them might dimerize in this way. So depending on the manner in which they dimerize could have huge structural implications, and also provide the kind of specificity that dictates which TLR will interact or dimerize with which other TLR. So the chemistry is important. Also, the mode of the dimerization at the TIR domain is also important. And for this, Shaila is going to present our attempts to model them as accurately as possible. And to follow this, there is this pink-colored region which we keep calling it as a BB loop. And the BB loop can be facing each other like that, or it's a little far away from each other, and the third one is somewhat similar to the second. However, Shaila is going to explain how we care for the dimer orientation when we model the system, especially the intercellular TIR domains. And then finally, and more bravely, Vikas Tiwari has attempted the full-length modeling of one of the TLRs called the TLR10, which is acquired less attention, but is known to bind to RNA. So with the full-length model available, he has gone forward to also model how it might interact with RNA, how it might be present in the lipid bilayer. After all, the transmembrane helix is in the lipid bilayer, yeah. And finally, how the mechanism of action might happen, how the substrate might be shared, and how the signal transduction may happen, and so on. So this is all part of the first story. First, you will hear Mahita's recording, and the second is Shaila will explain the TIR dimers, and thirdly, the full-length modeling of one of the TLRs, and the TLR10 will be presented by Vikas. So first, we will hear Mahita's recording. Please wait.
Hi, my name is Marita, and I am a postdoc at the La Jolla Institute of Immunology. I was PhD student in the lab from 2020 to 2017. And so in this talk, we will talk about the application of protein modeling to answer a biological question. You see an example of autoimmunity or threat with the protein involved in innate immune signal. Now, autoimmunity threat is important to prevent reconstituting activation. The attribute protein is actually completed the internal domain and the TIR domain, and they are separated by a protein region and the C-terminal disordered region. And it has been reported that the internal domain bound to the TIR domain as a mechanism of autoimmunity to prevent constitutive activation or truth. Now, the answer structure of the internal domain and the TIR domain, but we do not have the information of the entire protein. So, in order to get a model of the traits in the autoimmune state, what we did first was we did a blind talk between the internal domain and the TIR domain, and this was followed by guidance donkey. And then Ranku was the talk process, followed by validation and then simulation to assess the dream. At the time that the talkie and simulation actually suggests that the truth and terminal domain bound to the primitive region of the truth TIR domain, and the microsecond simulation highlights the extremity of the talk complex. And hydrogen interaction that hydrogen bonds the at the interface between the internal domain at the TIR domain. As you can see from the empty simulation, at this plot to show the occupancy of hydrogen bonds at the amount of time to the empty simulation. So based on this modality, we were able to actually propose a mechanism of activation from autoimmune to the adjuncted, and the lack of time I cannot explain about this mechanism, but if you are interested to know more, please refer to this paper in literature. And thank you for your time.
That was Mahita explaining about how the TRIF N and C terminal domains were modeled and the interactions were also modeled. Next, I would request Shaya Verma to explain the modeling of the TLR diamonds. Shaya.
So hello everyone. Today I'll be talking about variety in TIR domain formation. You all just heard about how the TRIF, which is one of the adapter protein, works in case of TIR, and how the TIR are important for the functioning. So TIR domains are a part of the toll-like receptor proteins. So these are various types which can be either on the plasma membrane or on the endosome membrane. And these either homodimerize, like in case of TLR 5, 10, 11, 6, 4, but also it can heterodimerize with different kind of unit like TLR 6 and TLR 2, TLR 2 and TLR 1 and so on. Also, if you look at the structure of toll-like receptor, it has an extracellular domain, a transmembrane domain, and a TIR intracellular domain. So what are the important regions of the intracellular TI domain, which are important for the dimer formation? If we look closely into these, there are something called as C-helix loop. As ma'am just mentioned, it can be dimerizing either in this way or in some other pattern. So the important factors are the C-helix or the BB loop shown in blue color over here, which is a loop-like region and help in the dimer formation. So going ahead, we just looked at how these different PDB, what are the different PDB models, which shows how the TIR interacts. So for some example, this is a TLR 10 dimer, which is from a crystal structure. And we can see the BB loop, which is in pink color, seem to play an important role in the dimerization. And it looks very symmetric if we consider both the monomers. The other type of TLR, which is TLR 6 TIR. So here if you see the BB loop seem to be far distance from each other as compared to TLR 10. And also this is also symmetric, but the regions are not same as in case of TLR 10, but there are different other helixes which are important for these kind of interaction. The another TIR containing protein is interleukin receptor protein. So they also have BB loop in their structure and a 2 TIR, which constitutes to the dimer formation. But in this interaction, the BB loops are seen to be far apart from each other. So to look for how these TIR domain interact in case of toll-like receptor, this is a work from the lab by Mahita and ma'am, where they have shown how TLR1 and TLR2 interacts, how TLR3 interacts, and how TLR4 interacts. So TLR10 being the closest in terms of interaction as per most of the experimental studies and also sequence identity. So the model was built, and what we found that the BB loop of both TLR1 and TLR2 plays a major role in the heterodimer formation. Also, in case of TLR3, again the BB loop, which is shown in the pink color over here, seems to be at the interface and leading to a lot of interaction in the dimer formation. Similarly, in case of TLR4 homodimer, again comes the BB loop, which is important for the dimer formation. So why these become important? Because if there is any mutation in the nearby region, it can affect the overall binding parameters between the monomer and thereby the downstream signaling. So to quote one of the examples from this paper, so there was the study where the communities in terms of the amino acid which are interacting between these two dimers were observed, and they found that few of the residues from each of the chain were important for interaction. But in case of a mutation, say P681 to H, which is in the baby loop, it changes the community pattern in case of TLR1 and TLR1 and TLR2 interaction. And because of such changes, the downstream signaling is being abrogated, and the TIR binding to the mid-88, which is the downstream partner, is stopped. Similarly, in case of TLR3, similar patterns were observed where there was a larger community involving 7 residues in the case of wild type, but in case of mutants, which is A795 to proline, the changes in the residue were leading to a community of 6 residue and also abrogating the downstream signaling. So looking at one of the TIR from TLR4, so let's see what are the different approaches we can take. So there can be a structure-based approach where we have different templates. So say this is based on a Galaxy web-based server. So where TIR domain, which are available from different toll-like receptor proteins, were used as template, and we found that this shows the most similar as the BB loop is seen to be as in the expected condition. Whereas in other templates like from TLR6, TLR1 and 2, the BB loops seem to be very far away, which doesn't satisfy the condition as these seems to be very interactive because of their functioning. There can be other approaches where we can look how two monomers can interact. So the other approach can be a Piper docking, which is via Schrodinger suit, and then later on validating the score through an in-house tool, which is doc score, to see what are the best possible confirmations how this BB loop can interact to make the dimer stable. The other can be an haddock approach, which is a energy-based approach using biochemical functions and even providing sufficient information of key residues which are important, but this may not always take in consideration the actual pose and give something which is really true as per the functioning. Here we see the BB loop comes together, but not in the same symmetrically opposite manner. So later, why these modeling becomes important? Because with the help of such modeling, we can look for the downstream partners and how any abrogation or any changes will obstruct, slides are not moving, sorry. Yeah, okay, yeah. So why these modeling become important? Because suppose this is the TIR domain, and then it interacts with the blue region, which is a tram dimer. And then this is a green region, which represents the TRIF protein. So this is one of the big complex of the TLR4 intracellular protein signaling. So through the models, we can understand what are the important sites which are involved in the functioning, say the BB loop of the tram TIR dimer, the BB loop of the TLR4 TIR dimer, and the BB loop of TRIF, and how these are playing key roles, and also some of the key residues which are there, and how these will help in the complex formation. Thank you. Over to you, ma'am.
Thank you, Shailya. So now I will request Vikas to briefly present the full-length modeling of TLR10.
Thank you, ma'am. And good afternoon, everyone. In this talk, I will be describing the structural modeling of full-length toll-like receptor 10. So in human, there are 10 different toll-like receptors, namely TLR1 to TLR10. And these TLRs recognize multiple pathogen-associated molecular patterns. And among these TLRs, TLR10 is the least characterized TLR. And this is the only TLR with unknown structure for the ligand binding domain, which is the extracellular domain. And also in case of all other TLRs as well, there is no full-length experimental structure available. So to answer this question, we employed computational technique of modeling. So to begin with, we first modeled the monomeric structure of TLR10 extracellular domain, and then by choosing the relevant templates of the dimeric structures, we built 4 different homodimers of TLR10 extracellular domain, which can be termed like closed model or the open model or models in between. And further, for modeling the transmembrane helix, we employed protein-protein docking to build the dimeric form of the transmembrane helix. And for the intracellular domain, which is TIR domain, the crystal structure is available. So by utilizing these domain dimeric structures, we finally built 4 different full-length models. And through energetic calculations, we observed that the closed model is energetically more favorable, which can be attributed to its elaborate interface. And also, we wanted to understand the domain dynamics, for which we subjected these static models to the molecular dynamic simulation in membrane aqueous environment. And by the end of the simulation, we observed that both extracellular domain and the intracellular domain tilt towards the membrane bilayer. Although both the domains show significant movement, the dimers do not dissociate throughout the simulation time, and we could identify multiple interacting residues between the protomers, which can be further used in the experimental validation. Next, we compared our model with the AlphaFold model. Here we can see the magenta one is the AlphaFold model, and when we superimpose the AlphaFold model with our model, the domain-wise superposition is good, but the domain organization in general is not good for the AlphaFold model, simply because when we put the lipid bilayer, it shows several clashes. And therefore, we went ahead with our model structures for further analysis. Next, recently it has been shown that TLR10 can recognize double-stranded RNA when it localizes to the endosome. And to understand the interaction between the RNA and TLR10, we did the RNA protein docking, and we could identify few conserved histidine residues which can interact with the double-stranded RNA as shown in these figures. We did this docking of RNA in the closed form of TLR10 model as well. And we found through MD simulation, we subjected this complex again to the MD simulation, and we found that the complex formed is stable in nature. Further, to understand the signal propagation, we built the dynamic network for the TLR10 RNA complex. And in this network analysis, we can find out the critical residues, which are important for intra-protein signal propagation. And also we can identify the optimal path between two given residues. So in the presence and in the absence of the RNA, if the optimal path length decreases or the number of suboptimal paths increases, that indicates that the signal propagation becomes more efficient. And we did this analysis for both open form and the closed form. And through this analysis, we can hypothesize that the open model can be the functional form of TLR10 as the signal propagation becomes more efficient upon RNA binding, while the closed form model can be the upper form, even though it is energetically more favorable, simply because the template used for modeling the closed form has been experimentally validated to be the upper form. So this is what we are proposing for the TLR10 full-length model. Thank you.
Great. Thank you, Vikas. Thank you, Shaila, and also we heard Mohita's presentation. Now, that was the first story, which is on the toll-like receptors, looking at how modeling can help us understand a bit better on autoinhibition, dimer formation, and how the full-length structure can help us understand a bit better about the mechanism of action. But the second story that we plan to present today that employs three-dimensional modeling as a tool. So what I was going to say is that the availability of three-dimensional models have been helpful for us to understand a bit more on the toll-like receptors. But now next, in the second story, we plan to present the similar sort of an approach, but on another membrane protein, which is an olfactory receptors. The mammalian olfactory receptors are quite distant from the G protein coupled receptors whose structures are available. So we could have used the G protein coupled receptors or the GPCRs as our start point for 3D modeling of the olfactory receptors in the mammalian genomes. That is still possible, although they are distantly related. However, the evolutionary drift is such that the functional motifs of the GPCRs, the classic motifs such as the NPXSY or DRY motifs, are not observed in the olfactory receptors. Nevertheless, with some difficulty, it was possible to model mammalian ORs starting from GPCRs. And since this is the case, and there are hundreds of ORs in genomes like the human genome and mouse genome, Harini in the lab, and together with a few more people, had set out to identify what sort of ORs can we commit for three-dimensional modeling. And they provided some kind of objective scoring scheme, that such as getting to know the sequence identity with the best GPCR so that we are getting the most reliable model, but also the length of the loop regions and so on. And then we finally committed to building 100 mammalian ORs. So we called in the title of the paper also, it said 100 ORs for the molecular biochemist or something nice like that. So Harini will present that part of the story, modeling mammalian, some of the mammalian olfactory receptors. On the other hand, the story for the insect ORs is completely different, because not only they don't see the GPCRs, even the topology is pretty different. The N-terminal will be facing the intracellular milieu of the cell and so on. We were at such a situation, however, as luck would have it, the cryo-EM structure of the co-receptor of the insect ORs became available. And what are these co-receptors? These are, as the name suggests, they always work with an OR by forming a heterodimer, and together they perform the signal transduction. And whereas the ligand binding is believed to be at the OR extracellular site, the signal transduction itself for the insect ORs happens at the co-receptor, co-receptor, and they act like ion channels, not like the typical GPCRs in any way. However, what Vikas Tiwari is going to present is his experience of trying to model the insect ORs from the insect OR structure, and also how the OR or co might themselves interact with each other. So this is what we will be hearing in the second story. First from Harini on the modeling of mammalian ORs, and secondly from Vikas on the modeling of insect OR structures. I would request Harini to present the first part of this second story.
Thank you, ma'am. Good afternoon, everyone. I'm Harini. I'm a postdoctoral fellow at Professor Raghuparanjit's lab at NCBS. I did my PhD in Professor Saurami's lab from 2010 to 2015. And it's a proud moment and it's a pleasure to be presenting in this 25 years of CAPS, the ongoing series of talks. I'll be talking about molecular modeling of olfactory receptors. So to give you a brief introduction, olfactory receptors, as ma'am just mentioned, belong to class A GPCR proteins, and they have a 7-transmembrane helical structure with an, in the mammalian ORs, it's an extracellular N-terminus and an intracellular C-terminus loop. In the insect olfactory receptors, especially the dipteran insects, it's an intracellular N-terminus and an extracellular C-terminus, suggesting that the topology is reversed. The intracellular signaling, however, is similar to GPCRs, where upon odorant binding, the G-alpha dissociates and converts ATP to cyclic AMP, which then propagates further ion channel activation at the membrane. So what we set out to do is to develop the structure in silico, using in silico homology modeling, develop the structure of 100 mammalian olfactory receptors. So to do so, we had to select a template that is a protein which is closely related to olfactory receptors and also has a 3-dimensional X-ray crystallographic structure in the PDB database. So we selected bovine rhodopsin because it was the sequence of a class A GPCR, which had close identity to olfactory receptors. We then would obtain an alignment for each olfactory receptor that we selected with the template and map the secondary structure regions, that is the 7-transmembrane helix on the alignment, which is seen here in the rainbow colors. A high identity at the secondary structure region will help us to get a good model at the end. We then used Modeler that was developed by Sally et al. In 2003 to obtain 20 different models of each olfactory receptor. And based on the energy values, we selected 5 best models with the least energy. We would then validate each of this model using Ramachandran map, which tells us the correctness of 5 sine angles in the model that we have developed. We also perform loop modeling for each model because, as I mentioned, olfactory receptors contain 3 intracellular and 3 extracellular loops, and each of this loop conformation is very important for the function of the protein. So we've used Modeler again for performing loop modeling for each of these models, energy minimized the best conformation, and selected the least energy structure. The complete structure with the transmembrane helices and the loops were then energy minimized, and the final model was obtained, and this model was used for further studies. We also attempted to model one of the insect olfactory receptor co-receptor, but the challenge here was that we did not find any template that had high identity in the PDB. However, for some of the transmembrane helices, that is 1, 2, 3, and 7, the bovine rhodopsin model, bovine rhodopsin template had high identity. However, for the other helices, that is TM4, 5, and 6, we found that the sequence identity was higher with potassium channel. And we also know from other studies that this receptor by itself can function as a channel at the membrane. So here we did a multiple template modeling, wherein we used 3 different templates to get the structure of the insect olfactory receptor, about which Vikas will be discussing further. So with the 100 models that we generated, I'll just explain few applications of what we did with those models. So the first one was with the mouse olfactory receptor, where we worked with Professor Gromeha at IIT Chennai, who helped us to understand the correlation between amino acid property and the EC50 values of odorant binding to a receptor, and how this information of structure can help in validating these results. So the correlation when only the sequence information and the EC50 values were used was 0.63. However, when we had the structure information about olfactory receptor, the correlation between the amino acid properties and EC50 values increased to 0.81, which suggests that having a structure is quite important to understand the function of olfactory receptors. So I've just marked those residues whose mutations were studied on the structure here. We also went ahead to check the dimer interface of olfactory receptors because we know that most of the olfactory receptors either function as homo-oligomers or hetero-oligomers. In this work, which was done with in collaboration with Professor Namato at Japan, we identified for all the 100 olfactory receptors the dimer interface. So what you can see at the center is a phylogenetic tree of all the mammalian olfactory receptors, and the numbers on the side indicate the ones that we have modeled from each cluster. And you can see that, for example, in the cluster 1, that the dimer interface that has been predicted are different regions of the transmembrane helices, and some are in the loops, and it is not constant or similar across olfactory receptors which share high sequence identity, that is, those which belong to the same cluster. So this again further confirms the fact that every olfactory receptor has a different function and can bind different set of odorants, even though they belong to the same cluster or they're sequentially similar. All of this data that we have obtained has been deposited in a database of olfactory receptors, which is accessible to public, where you can have the sequence alignment between the olfactory receptor and its template, the structure that was obtained, the validation of the structure by Ramchandran plot, and the conservation of residues mapped on the sequence as well as the structure. Last, we did a brave thing where we selected about 160 odorants and did an induced fit docking with 10 pairs of mammalian receptors, human and mouse, which shared high sequence identity. And we obtained a heat map that shows us that what is the range of interaction or the energy of interaction between a receptor and an odorant. So we have now a data set where we know what odorants these receptors can interact with. We validated our study by using the known interactions. For example, as you can see here, we used some odorants that were very specific to insect odorant receptors, and we see that their energy of binding is quite low, suggesting that mammalian olfactory receptors do not bind the same odorants as the insect olfactory receptors. These are some of other examples where eugenol, eudecanol, and androsteroid was used to show that the receptors that are known to bind these odorants do pick up the same odorants with high energy values in our analysis. So this data as well is available for further studies. Thank you. And congratulations to CAPS.
Thank you, Bharani. Yes, it was indeed a very brave attempt to do this sort of a mini virtual screening. And I remember from the very first discussion, we used to have this heat map. And I'm really happy that you made all this happen, even as the structural information was quite limited at that time. And next, I request Vikas to present the second part of the second story, which is to perform three-dimensional modeling of insect ORs and just one of the insect ORs and ortho, the co-receptor, and how they two might interact. Because please.
Thank you, ma'am. And for this talk, I will be discussing about the structural modeling of odorant receptor from mosquito Aedes aegypti. So the reason for selecting OR4 is that this particular OR has been shown to impart human host preference to the mosquito, and all these ORs require a co-receptor called ORCO, unlike mammalian ORs, for their proper functioning. So in this case, we have selected the co-receptor ORCO and OR4 for structural studies. So to begin with, we started with the finding the homologous templates for modeling the ORCO structure. So the only available structure at that time was for the ORCO structure from the fig wasp. It is a homotetramer ion channel in the closed form. So from this tetrameric assembly, the monomer can be selected as a template. We aligned our query sequence to this template, and as can be seen from this alignment, only for the intracellular loop 2 region, which was not resolved properly in the template structure, we did the this part of modeling separately, while the rest of the structure was built using this template. The final structure had similar 7 transmembrane helices marked in different colors. Later, to refine this model further, we subjected it to short MD simulation in multiple runs. And we observed that the structure at the end of the simulation had no residue in the disallowed region of Ramachandran plot. And the residues in the modeled intracellular loop and also the extracellular loop 2 regions had high root mean square fluctuations than the transmembrane helix regions. In case of orco, which is a conserved molecule across insect orders, we can identify important residues like the triad of polar residues or the hydrophobic residues which form the gate in case of the ion channel. And now once we have this final refined model for the orco, we can use this for screening multiple natural compounds for potential repellent molecule identification, or this can also be used for building the complex structure for orco and OR4. So this particular fundamental question of like how different ORs interact with orco has remained elusive till date. So we employed a similar strategy for building the model of OR4 as well. Although in this case, the sequence identity is very less between the template and the query. But experimentally, it has been observed that the overall architecture of OR4 and ORCO remains same. Therefore, we use the same template. And in this case, also the final refined model had no residue in the disallowed region of Ramachandran plot. So as we mentioned that ORs belong to family which have very diverse set of sequences. So we wanted to understand if there are any particular structural element which remain conserved in the OR4. For this, we extracted all the OR4 sequences and we did the multiple sequence alignment, removed the outliers, and we repeated this exercise. And finally, we had around 100 OR sequences, and we did the conservation analysis using conserve. Once we have the structure as well as the conservation scores, we can map it to the structure, and we identified that the 7th transmembrane helix of OR4 is relatively more conserved compared to the rest of the structure. And similar was true for the orco, even though the orco is highly conserved across multiple insect orders. For building the complex of OR4 and the orco, we employed multiple protein-protein docking tools, including AlphaFold multimer, and also HADDOC, which requires some experimental information for guiding the docking protocol, and HDoc, which generally does the docking using in a blind manner or in a global manner. So once we have these docked complexes, we can employ the topological information as well as the evolutionary information to filter the wrong poses. And in all these docking approaches, we identified that the 7th transmembrane helix, which is found to be conserved, is also involved in the interaction with the co-receptors. So from these docking studies, we conclude that the orco can interact with a diverse set of ORs through the 7th transmembrane helix and also residues from the intracellular loop 3. Thank you.
Thank you. Thank you, Vikas. I must say that much of the sort of modeling that we have explained in the first 2 stories have not been easy. First of all, these are membrane proteins, and second of all, the sequence identity is pretty low. And third of all, we don't always have the luxury of a single template. So much of these had to be done by means of very large number of trials and iterations. And I must say that the time that I had spent as a postdoctoral fellow in Professor Tom Blundell's group had been quite valuable to work in borderline sequence identity. And sometimes we have also worked with cases where the sequence identity is as low as less than 10%, which might be between 2 random proteins that get aligned as well. Right. Now, what we will do is to switch gears, and then we would now, for a change, next look at a globular protein. I think it is all right to call it as a globular protein because what we have is a system, a gene called CSRP3, and that stands for cysteine serine rich protein. And as the name suggests, it has a large number of cysteines and serines in its amino acid sequence. And not only that, it is made up of 2 limb domains, and they are connected by a fairly disordered region. So here again, there are challenges in 3-dimensional modeling. But what we are hoping to showcase today is after such a 3-dimensional model of individual limb domains, how can we use 3D models to better understand or provide a structural rationale for deleterious mutations, because many mutations have been mapped onto this gene, the CSRP gene, and more to be more specific, they get mapped onto the first limb domain, the limb 1 domain. And the Pankaj of my lab had noticed that many of the cardiomyopathy mutations, whether it is a hypertrophic or dilated cardiomyopathy, are getting mapped more on the first domain. And not only that, he went a step ahead and made some very imaginative studies, which is he decided to perform a virtual saturation mutagenesis, which is at every alignment position, he would mutate that residue virtually to all other 19 amino acids except 7. And therefore was able to provide a mutational landscape, which kind of provides a computational picture about where mutations can be tolerated and where mutations just cannot be tolerated. And all those which are shown in this dark color means that these are the positions where mutations are not possible to almost any other amino acid. And these are very rigid. So next what we are going to hear is the recording by Pankaj and his narration about his experience understanding the mutations and performing the virtual saturation mutagenesis. Please stay on.
Good afternoon, everyone. In next couple of minutes, I'll be talking about our recent structural work on a very interesting protein known as CSRP3. Cysteine and glycine-rich protein 3, or CSRP3, is a dual-role mechanism sensor. It is very important for the sarcomere stability. At the same time, it is also important for the tissue remodeling, where it shuttles from the cytoplasm to the nucleus and acts as the cofactor for the myogenic transcription factors. Mutations in this particular protein are known to cause the heart muscle diseases, known as dilated cardiomyopathy and hypertrophic cardiomyopathy. The very first question we ask, where are these disease-causing mutations localized in the CSRP3 protein? For this, we surveyed all the known mutations in the CSRP3 and mapped to the sequence of the CSRP3. And we observed that most of the disease-causing mutations are localized in limb domains. There are two limb domains in the CSRP3, N-terminal and C-terminal, and these are connected by highly disordered linkage region. And N-terminal limb domain harbors most of the disease-causing mutations. The next question we ask, how these disease-causing mutations are affecting the limb domain. For this, we carried out the in silico point mutagenesis, where we substitute the original amino acid with the disease-causing ones and measure the delta delta G, or change in Gibbs free energy. And our analysis showed that majority of the disease-causing mutations are destabilizing in nature. The next question we asked, is there any position which can accept any possible substitution? At the same time, is there any position which is intolerant to any substitution? For this, we carried out in silico saturation mutagenesis, where we substitute all the positions in the limb domain with all the possible 19 amino acid substitutions and measure the delta delta G. In this way, we get the mutational map. And this map showed that clearly there are positions which are intolerant to any substitution. At the same time, there are positions which can accept any substitution and don't affect the structure. To validate this map, we selected the 44th position, where you can see that all the amino acid substitutions are destabilizing, marked as red, except the methionine one. And previously, we had found that there is a disease-causing mutation leucine to proline substitution at the 44th position, which is marked in the star. So we selected this disease-causing variant, the neutral one, and the wild type for further analysis, and we carried out the MD simulation analysis. Our MD simulation analysis showed that disease-causing variant has significantly different trajectory compared to the wild type and the neutral one. In this panel, we see that where we plot the secondary structure throughout the trajectory, wild type and well as the neutral variant show similar pattern of secondary structures. At the same time, disease-causing variant shows the totally different pattern where the extended beta strand as well as the isolated trees are lost throughout the trajectory. If we zoom at the structure, we see that the 44 positions which interacts with the long range interaction with the phenylalanine 35 in the wild type as well as the neutral, it is lost completely in the disease causing variant. In the interest of time, I won't be presenting the other analysis, but in short, we were able to show that this particular pipeline can be utilized for in silico rapid analysis of the point mutations. Thank you.
That was great. Okay, so as a result, what we have heard today are three diverse stories, and I hope you enjoyed listening to them as much as we enjoyed presenting them to you. And the first of the story was on toll-like receptors, and there were three parts to it. The first part was presented by Mahita, who's currently a postdoctoral fellow in the Institute for Immunology in California. And the second part of the story, which was on the TIR domain, was presented by Shailya Verma, who's a current PhD student. And the third part was on the full length of the TLR10, which was presented by Vikas Tiwari, who's also a student presently in the lab. And the second story was on olfactory receptors. The mammalian olfactory receptor part was presented by Dr. Harini Krishnan, who's currently a postdoctoral fellow in Raghupat Injet's lab. And the second part of the second story was on the insect olfactory receptors, was presented by Vikas Tiwari again, who is currently a PhD student in our lab. And thirdly, we had briefly presented the experience of understanding mutations on the CSRP3 gene, and in particular the cardiomyopathy genes. And this was presented by Pankaj Chauhan, who has very recently defended his thesis and also obtained his PhD degree. So he's currently a postdoctoral fellow funded by the IVAB grant, Institute grant. So with this, I stop my part of the narrations as well. And we got a number of very interesting questions. Thank you to the audience for thinking and asking them. And I would also next like to request two people to conclude today's seminar. First, Shafi, whom I would request can present the list of papers or publications that are relevant to today's discussion. And he would also announce the topic for the next talk that is due to happen next Thursday. And since we are finishing the structural theme, we have had 4 talks so far. So I would next request Advait to provide a consolidation picture, the timeframes for the projects, time for the people who are present, and also when the publications have emerged. With this, I would request Shafi and then next Advait to present their part of the narrative. Shafi, please.
Hello everyone. So these are the relevant publications related to today's talk. And first session we had toll-like receptors modeling, and we have 3 Mahitas and 4 other papers regarding toll-like receptors modeling. And we have a recent paper from Vikas Tiwari talking about LR10 modeling and analysis. And in the next section we talked about olfactory receptors modeling, and we have first 2 papers from Harini et al. And we have the third paper that is talking about DOR database, and we have another recent paper from Vikas Tiwari regarding olfactory receptors. And the third section we have CSRP3 from Pankaj Chauhan, and he talked about cardiomyopathy mutations in CSRP3 protein. So for next week in CAPS talks, we have the last set of talk in the structure team. We have titled this protein three-dimensional models are hidden jewels for protein prediction. Please state it.
Next, I would request Advait to present a consolidation of the talks that were presented in the structure theme. Advait, please.
Okay, thank you, ma'am. So we finished the structure theme today, and I'll just take you all to the journey of how we all had all these 4 talks discussing the protein structures. Okay, so we started on 9th of March with the first talk on structure theme, where we first started with the second structural conservation, where we discussed about an algorithm called as CUSP, where key players were Sandhya Sankaran and Ishita Math, and we also had multiple publications coming out of this work. Next, we talked about protein-protein interactions, and basically an algorithm which was developed to score the docking poses, which is called as dock score, where Sony worked on it, and currently it is being pursued by Revati. Next, we also discussed about coil check and another algorithm for coil proteins energy interaction. And there are several players in this, such as Anirban Bhaduri, Vikram Alwas, Divya Samala Devi, and Margaret Sunita. And finally, that day, we also discussed about another algorithm, which is called as PP check, which involves Prashant Shimte, Anshul Supwar, and Uman Matthew. And this is an algorithm to calculate the electrostatic potentials in the protein-protein interaction. So next, the second talk in the structure theme happened on 16th of March, where we discussed about protein domain identification, starting with an algorithm called as DIAL, and the subsequent database generation called as DDBase, which is dial-derived domain database. And we discussed work on DIAL as well as DDBase, where key members were ma'am, Professor Saudamini, and Vinayagam, and Dr. Meenakshi Iyer. And next, we also talked about our lab's interest in domain swapping work, where we have looked at domain swapping extensively and also created an algorithm database called as 3D swap. And the key members were Pugalanti, Shamir Khader, Prashant Shimte, and Atul Upadhyay. So these were first 2 sessions of the structure theme. The third session started with disulfide rich proteins, where we first talked about MODI, which was originally the PhD work of Professor Saudamini, and then how it grew to a disulfide containing disulfide rich proteins called as DSD base, the database, which went into several transitions from 2004, then 2014, where version 1.2, and to the 2022 version, where second version was released. And key players in this work were Vinayagam, Pugalanti Rajesh, and Professor Saudamini. And then in the 1.2 version, were Soumya Indra Kumar, Murugavel Pavalam, Professor Srinivasan, and Professor Saudamini. And in the latest version, which is the 2002 version, Neha Kalmankar, Murugavel Pavalam, Soumya Indra Kumar, Professor Saudamini, and Professor Srinivasan. Then we talked about higher order residue interaction, which is in form of a tool called as HORI, and there were 2 versions of it. The 2010 version, where the team members were Sundarmurthy, Rashi Srinivasan, Shamir Khader, and Professor Saudamini, and the current ongoing version, where Sarthak is also looking at it. And we also discussed about another algorithm for structure validation called as Harmony, where Pugalanti, Shamir Khader, Professor Srinivasan, and Saudamini were involved in that. And then we talked about our lab's flagship database, whose inception was even before the lab started. So it has a nice journey with as the lab grew. So the database also grew a lot, where it started in 1998 when it was called as Campus, while Professor Sadami was doing her postdoctoral studies in Cambridge. And then it changed into or metamorphosized into PAS2 and grew up while we are updating it till today with several versions coming from different years 2002, 2004, 8, 12, so on until the latest version, which is 2022. And there are so many players involved in PAS2 work. I'll go to the last talk, which happened today, where we covered several protein structure modeling stories, starting with toll-like receptors, where key members are Mahita, Vikas, and Shailya. You heard about their protein modeling exercises today. And next we heard from Harini and Vikas about olfactory receptors. And finally, we heard from Pankaj about a protein CSRP3 involved in cardiomyopathy and also saturation mutagenesis experiments done in this regard. So this is where we conclude our structure theme. And from next week, we'll be starting with the function theme. And as you saw, the title for the next talk on Thursday, 6th of April. Please stay tuned for this.
Yeah, thank you.
Over to you,
Advait. Thank you, Advait, for a very beautiful consolidation. But as if all this was not exciting enough, we now have a jewel in the crown because you can see Anirban Baduri has joined online. And I want to just yeah, hello Anirban. Yeah, it's great that you could join us for this discussion. And for the audience, let me just remind you that Anirban has been kind of occupying both the sequence and structure theme in many seminars. He was there. I talked about his work on the spatially interacting motifs, and then on GenDis, and then on IMPA, which is to use these motifs as in sequence searches. And then he had also participated in PAS2. He had worked on the preliminary parts of CoilCheck, as well as worked majorly on phosphatases. Plus, he has also contributed to the alpha-beta hydrolase story that gave birth to the HORI project that I discussed. So he has been a very imaginative and helpful colleague. And currently, I think we've been unfortunate that the company has absorbed him. I want to say, Nirvan, say a few words, maybe.
Sure. Thanks, Adamini, and always a pleasure to come back and talk. And I should mention two things. I mean, so Adamini gave a huge list of things that I did. But it wouldn't have been possible had she not been there and she not been flexible to whatever I was doing. So, I mean, the fact that I got that opportunity to work, I think a lot of credit goes to Sudhamini. But yes, I mean, it was really fun days, and I'm sure you guys also have a lot of fun even today. But one advantage that at least I had was it was the early days of the lab, and we were trying to build things. So at least I cherished the moment that I was there, and whatever it is, though Sudhamini says that I've been dropped by companies, but I still love visiting academic institutes. And I need to confess about one thing, that I have not come to institutes for quite some time, but that's because right now I'm in Pune, but I keep coming to Bangalore, and whenever I get there back, I promise I'll visit institutes.
Yeah, that's great. I mean, once I do visit NCBS and take some of your, things that you could maybe give a seminar.
Absolutely, would love to.
Whatever the company permits.
Yeah, absolutely. I mean, that's also part of the job. So would love to share whatever we like.
And yeah, thanks to the audience for your patient hearing. And do take care, because we will meet next week, same time, next Thursday at 3 p.m. Bye for now.
Bye, everyone.
I would request the panelists to stay on for a little while. But I have a question for Vikas. Thanks for your work on modeling insect co-receptors. Have you verified dimer interface difference between the newly modeled dimer ortho and the AlphaFold model? Cavity difference, maybe. Yeah, this is interesting because we pursued. Yeah, so Vikas is answering and he has already answered, I think. Okay, I think Vikas might answer it live because it takes time to type. So he will answer it for you.
So in case of complex modeling, we have not compared the AlphaFold complex with the complex generated from the other docking tools.
Yeah,
hope
we
that answers the
mainly
question.
use that to see how much they are different from each other in an overall sense, how the model quality looks like, the orientations and so on, but we not kind of compare the cavity, the dimer interface between various tools. It's an interesting question. Yeah, I think we can pursue it. Thank you so much. Thank you, Rajesh.